Recently, monoclonal antibodies have been making some development in the vet field, i.e. animal science. These monoclonal antibodies have been used to treat various diseases and conditions such as allergies, dermatitis, osteoarthritis, and most recently, heartburn. To understand the significance of these developments, it's important to know what monoclonal antibodies are and the immunological science behind them. So first off, when you get sick, it is caused by an invasion of pathogens, and each pathogen carries antigens. And antigens, when inside the body, trigger what is known as an immune response. In an immune response, the body releases various white blood cells, one of which is called the B cell. The B cell's purpose in the immune system, once it comes in contact with the antigen, is to release protein structures called antibodies. Antibodies, when released, then attach themselves to the antigens and destroy, disrupt, or neutralize these antigens so that they're no longer a threat to the body. This relationship between antibodies and antigens is what allows the body to recover when it gets sick. However, it's a bit more complicated than that. So remember how I said B cells release antibodies which attach themselves to antigens? Well, not just any antibody will react to any antigen. Every B cell carries specific antibodies which will only react to a specific antigen. To make this easier, try imagining it in terms of locks and keys. Let's say we have a yellow lock, an antigen, and a yellow key, an antibody. And let's say we have a blue lock, a different antigen, and a blue key, a different antibody. The yellow key or antibody can only open the yellow lock or antigen. Similarly, the blue key, representing an antibody, can only open the blue lock, antigen. The blue key can't open the yellow lock, and the yellow key can't open the blue lock. How does this relate to monoclonal antibodies? Well, monoclonal antibodies are just laboratory-produced imitations of your natural antibodies that are produced to fight against antigens. So, when treating an antigen, scientists will dissect the antigen and create the corresponding antibody. Monoclonal antibodies have been used in human medicine since the 1980s. It isn't until 30 years later that we've seen the benefits of these antibodies in vet science. And this is because, as I said, B cells are unique to a specific kind of antigen. So you can imagine it's been a little hard to develop the right antibody for these animal diseases. However, now we've got the ball rolling, and we've seen how monoclonal antibodies can revolutionize vet science, the only question left is, what next?